And we are recording. So welcome. I'm glad everyone's here. And we're going to be talking a little bit about publishing from a cure. One thing I do want to note at the start is we're not really changing what we want to do with the MCC. We're not changing the overall ideas. This is just an extension of what you can do. And so we're not, again, we're not expecting major changes. We're just offering this as a extra activity at the end of your cure if you want to go through with it. So uh, and I know Joe would want me to mention this for all those who are here and those who might be watching the recording later, that the professional development opportunity this summer, if you're interested in doing that. I didn't have a better picture, Joe, sorry. But at least go to the website, get some information, reach out to Joe or Amy or whoever you can find who has details about this, and it should be popping into the Slack, I assume, another month or so at the latest. So, yeah. And so the informal plan for this session and the future ones here is to be starting conversations about the spring and about different projects that may be going on, and to maybe potentially identify resources and gaps that we've got that we can do to promote collaboration and lead towards publications later this year, uh, later next year, I should say, since we're close to the end of the year, and early the following year. So today's conversation just is opening that idea. And then in January, we're going to start trying to assemble some working teams that are going to take data from their cure to a publication, looking at common areas and interests, compatible timelines, hopefully, and then also, well, as we're doing that, beginning to find resources, whether it's instrumentation or additional support, or maybe you need help publishing because you haven't done it a lot. You want someone who can look at your cover letter or someone who can help you choose the journal. So this is supposed to be the idea is that we're faculty helping other faculty and that all boats rise together. So uh, don't think that we are trying to, again, change what you're doing. The idea is that we are trying to promote each other's science and the quality that we have here. We're hoping that some of the people may be at ASB&B. We can have a meetup of some of the teams and begin to develop the collaborations further. And then during the spring, collect data, and then May, June, start up some more meetups to start the summer data scramble, where everyone's trying to collect data in that 6 to 10 weeks that we have during the summer. And then also we can provide further resources if needed. And then hopefully by early September, we're beginning the idea, beginning the outlining, beginning the draft of these papers so that we can get them in process. So again, today is really the beginning of hopefully some future meetings to begin collaborations, begin developing publications, and to move this whole group forward as a whole, and to add on to the quality of the cures that we already have in the system. So I thought I'd start actually with sort of a story of one of the early papers I did, and where we purified this protein called VIPF. It was a protein from Legionella. We purified it and did some structural work on it and looked at the size and did some modeling. We did some small angle X-ray scattering, and we also um, did some substrate identification. We just found that it, or described that it had an acetyl-CoA binding motif, and then found the substrate to be chloramphenicol. And then we found that each domain was a chloramphenicol acetyltransferase domain, which was pretty cool. And the reason I'm highlighting this paper is because several of these figures come out of cure data, in that some of the purification work was done by students in my cure, and we actually used one of their gels because it was a really nice one. The initial modeling of this structure from the sequence was done in the cure, and this is in the pre-AlphaFold days, so this is when modeling of this type of protein took sometimes days or weeks to refine, and so we did this as a part of a lecture project to look at the structure. And then the initial screening and identification that this protein bound to acetyl-CoA, and then finding that it could modify chloramphenicol, that was also done as part of an instructional lab. So half the figures in this paper really came from students who were working in an instructional laboratory. And so this is an exciting paper, but really is one of the reinforcements for me personally that cures can be ways to move science forward, and we can use those to get data that then becomes publishable later on. When we think about the scientific process, going from observations and investigations, come up with that preliminary idea to developing that hypothesis and then figuring out our experiments and analyzing it, cures are really good at supporting these early steps here. And towards the end of the semester, everyone's getting tired and we may just do a lab report and we don't really get to the point where we're taking that data and communicating externally. And this is sometimes a challenge because sometimes the data aren't that great, or it's incomplete, or the story's not fully there, or there's lots of challenges to taking cure data and getting it out to that communication phase. And so this is where some of us are thinking about how can we facilitate this process. And so this is again where we're starting today is can we push this further and really focus on this communication phase? Because yeah, as someone told me, you can't change the world if no one sees your data. So we want to get that data out there because we know that it's being generated across the nation in these wonderful MCC cures. So why publish? Well, the reason I put forward is we want to move the field forward. We're making a lot of mutations in MDH. We're looking at specificity. We're generating models. We're doing a lot of good work, and we want to get that out there so that the field can move forward. This also can be external validation or recognition of your and your students' skills in science. And this can be useful for promoting undergraduates to move on to the next phase of their career. I mean, there are hundreds of thousands of undergraduates graduating who have a 3.4 GPA. If they can get their name on a paper or some sort of external dissemination of their work, that can help them stand out. It also helps us and our students can connect with the scientific community. They can go out and present a poster or really get out there and meet others who may be able to help them in their future career. And it can help their communication skills and help them develop as people who can convey complicated ideas to the layperson. And it also sometimes publications are required for promotion and or tenure, depending on what type of institution you're at. So these are just some of the reasons. There are others out there, but these are the ones that I thought of as I was coming up with. Why should we be doing this? Why put in this extra effort? So if we are building out our paper or our poster, most MCC cures have some sort of enzyme purification step, activity assays, and there may be a model of a mutant versus a wild type that's going on there. We want to be able to move that towards a poster or paper. And one of the easiest ways to do that is to do some sort of kinetic analysis where you calculate Vmax and Km and maybe even Ki if you're looking at oxaloacetate. And then that can be your poster. And certainly there are opportunities where that can be a paper. I know that there's a paper that was recently published from Varuni and Ellis and a few others that has looked at a series of mutants in watermelon MDH. And it was mostly kinetic analysis, and that was the whole paper. So this can be a complete story with just the initial work that the cure does, and then adding on a little bit of kinetic analysis later, whether that's done by the students in the cure or summer students, or maybe even by you. There are other things that could be added. You could add on some ligand docking or molecular dynamics if you wanted. If you have access to it, you could do stability measurements and look at how the mutant affects structure. You could do more structural studies or modeling. I know we have some very talented cellular and molecular biologists in the MCC. You may just jump to cellular types of experiments. All of these together can get you to this end goal of getting that information out there and getting your poster or paper presented. So as we're thinking about this, don't think that what we do in the cure isn't enough. It is most of the way there. If you add a little bit of kinetic analysis, that gets you there. And then these other things are really just icing or garnish to make it look a little fancier and improve the depth of the story. But the story can still go just with the kinetic analysis. Just to show some alternative workflows, because I know that there are some out there like me who don't typically do a lot of mutant studies. When I'm looking at my biochemistry lab, we're purifying a protein and we're often looking at chemicals that could be inhibitors or activators of MDH. And so we frequently are doing inhibition kinetics and docking things to proteins and going that way. Um, I do a general chemistry lab where we are doing something very similar, and we're looking more at structures and thermodynamic properties. And so These are other ways that you may want to have your cure structured if you don't want to do mutants, but you may want to do something else that's related to MDH. So as we're thinking about how to get started and move our project in the spring or fall or summer, whenever we're teaching it, to this idea of what do we need to do to start this process of getting this paper going. The first thing to do is really figure out your question, your focus area. This could be something of your own personal interest. It could be the loop mutants, or it could be the phosphomutants or something else. And then decide, are you going to go alone on this, or is this going to be a collaborative project? The MCC has a wealth of people in it who can help or who often are doing the same thing. And so if we can find others who are doing similar types of studies, maybe you can divide the labor up a little bit. And one of the key things I recommend doing is have a discussion early about what is everyone doing, who is going to be sort of the corresponding author and the leader, and get things organized. You can have multiple corresponding authors, but really you want to figure out the roles of everyone early on so there's no misunderstandings and there's good communication from the beginning. And then figure out, is everyone doing the same experiments? Certainly you can have replication going on where each school that's participating is doing the same sets of mutants and doing the same types of experiments. If someone wanted to work with me and do something with my lecture group, maybe my lecture group is doing modeling and molecular dynamics and docking and things like that. And the person who's doing the laboratory side is doing all the kinetic experiments. There are lots of ways for this can work, but we want to have that conversation early. Then figure out their timeline. When are tasks getting done? Is this something that needs to get done sooner than later because of a promotion and tenure decision that's pending, or a grant application, or something like that? And then always be aware that changes will need to be made. And so good communication and having a system where you know you can reach out and get a hold of your team in a reasonable time and have a good, honest discussion is always key to do. Um, if you're looking at potential collaborators, think about the MCC, who else is working on this, and trying to go from there. If you need instrumentation, two places that immediately come to mind are certainly University of San Diego, which has a lot of instrumentation and people who know how to run that instrumentation. I'm at JMU. We have a lot of instrumentation as well, and I know that there are a lot of MCC folks that are within a couple hours of JMU. If you'd want to use any of our instrumentation, we're happy to have you uh, come on site, or you can ship me samples, or we'll meet halfway or something like that. And I know that there are other schools as well, but these are the two that came to mind easily. But if you need something, reach out and let's see who has it or who has access even. And that can be a way to start as well. So don't limit yourself to, I can only do what I have here on my campus. The nice thing about being in this community is we have lots of things and we can share these instrumentation and opportunities and ideas to make everything better for everyone. All right. When we're publishing a paper, we want to think about the timeline and how long it's realistically going to take. So I'm going to give you sort of a pessimistic timeline and we can go from there. So let's say that we're doing a cure where we're looking at 4 different interesting mutants. So January to May is when you're getting things together and you're teaching the cure. You're collecting that initial data from the students who are participating. Right about June or so is when many of the summer sessions start for a lot of us. And this is when you want to start repeating purifications, kinetic experiments, maybe other experiments. And again, this is sometimes will be research students. It might be you, it might be collaborators, lots of ways it can work. August to December, start outlining, writing, maybe revising a draft. I know we're all busy and we do a lot of teaching and that can be difficult to write, but try to set aside a little time and you'll be surprised how quickly papers can come together if you've got a good story. A year later now, we are now submitting the paper to the journal. And we'll talk about some options in a moment. Hopefully you receive your reviews within 6 weeks or so, February or March. You revise them and add experiments if needed. Hopefully you don't have a reviewer number 2 who wants you to solve a cryo-EM structure or something like that. Hopefully you get a reviewer 2 who sees what you did and appreciates your science. Hopefully by May of that second year, you're ready to resubmit, and then it gets accepted May or June. You get your proofs mid-July, and then it's hopefully posted online by August or September now. So we're looking at 18 months or so from starting the project, getting it together to actually having it be done. And there's like some assumptions in there that everything works and the story is clean and everything like that. And also some assumptions about how long the journal process takes. But this is not a bad timeline for a good project to work and how it could go for you. So be thinking about this as you're thinking about, oh, I want to publish a paper. It may not happen next week. It's something that you need to have some thought into. It's going to take me 18 months or so sometimes to pull this off. So here's how I'm going to do things in the spring, just as an example, another example of how I'm going to structure mine. So in the spring, I have a biochem lecture that has 70 students in it. And then I have three lab sections, which have roughly 60 students. And I do want to point out that these 60 students are in this lecture. So it's very much an overlapping population. And so I can combine these two courses together and expand the quality and the depth of the science I'm going to do. So my project is going to be looking at mutants, something I don't do frequently, but we're going to do it this time because I've got a student who's really interested in these disease-linked variants of human MDH2. There's maybe 8 or 10 of them that have been where the evidence is pretty good that it's a direct link between MDH dysfunction and the disease itself, but the molecular basis for it isn't clear. And so our goal is to look at the molecular basis for these human MDH2 mutants. In my lecture, my team will be looking at, they're going to modeling this variant structure, they'll dock some substrates, they'll do molecular dynamics, and then they're actually going to deposit their data into GitHub. And so this is going to be a way for them individually to get their information out and to create a record that's connected to them of the data that they're doing. In lab, I've already got the clones. I don't need to make them. So they're going to be purifying these variants. They'll be looking at protein stability using circular dichroism, and they're going to measure the enzyme activity. And they'll make a lab report, and I'm not sure yet if I'll have them combine the data in GitHub, or we'll figure that out. Then I've got two research students who during the summer are going to reproduce a lot of this work and then hopefully start writing the paper. And then I aim to submit by September 1st. It's a lot shorter than what I had put on the previous slide there, but I'm pretty optimistic that I can pull it off. And so this is my goal, is to be dumping this paper off to a journal around September 1st, assuming all goes well. So again, this is how I'm organizing my thoughts and preparing to do my lab and my project to get it to a paper. Few more definitions and then we'll go to more of a general discussion. So, what does a corresponding author do? I mentioned this earlier that you should figure out who's corresponding or going to be the responsible party for it. And the main job of the corresponding author is to manage the submission to the journal and be the primary contact between the journal and the research team. They're going to be generally, they organize the writing and the editing, they write the cover letters, they're doing the final formatting of the text and the figures, and handling all of this stuff. So this is why identifying who this person's is early is helpful, is because they are the person who's got to pull a lot of the stuff together at the very end. And so they're going to need a lot of information from everyone. Now, again, you can have multiple corresponding authors and you can have a team do it, but you really want to have one person who's sort of in that role of making sure that all the boxes are checked before you submit it off to the journal. And I know that not a lot of people coming out of their graduate career and their postdoc had an opportunity to do this. And so if you haven't done a lot of publishing in your career, you may not have a lot of experience being corresponding author, if at all. And so if you have questions about how to be corresponding author in some of these things, reach out. And can be one of the things where those of us in the MCC who have published a little bit more can certainly support you and help make you successful in this effort. I don't want something like this where you just want someone to look over all of your materials before you submit it. I don't want that to stop someone from publishing. I'd rather take a couple of hours and work with that person to get it all done than to have them not get a paper out that really is a good paper. Um, which journals are cheap to publish in? A lot of journals are going to article processing charges, and these are fees that the journal charges the authors. Some journals are free-ish, and some journals are well over $1,000. I think some of the nature journals are in the $5,000 to $10,000 range. So we're looking at a pretty hefty fee to publish. Um, a couple of these journals listed here: Proteins, Structure, Function, Bioinformatics, ACS Biochemistry, and Micropublications Biology. Their publication fees tend to be on the lower side. So if you're not someone who has grant money or institutional support for publications, you may look at these journals as places where you can get that information out. Now, there is also something called a preprint, which is a non-peer-reviewed version of your article, but you can put it out there. It'll have a digital identifier, a DOI number. You can use it as a work product for some grant agencies as well. BioArchive is a good one for MDH papers. And usually you can submit it. It gets screened just for being appropriate, and then it gets posted online in a week, and it is open access. So this is another way to get your information out sooner than later, and before it gets published in a peer-reviewed journal even. I do want to highlight micropublication biology because I've had reasonable experience with it. This is the recent figure from the recent work that my lab did, measuring NADH concentration using BCA dye. And this actually came out of a cure as well, this figure here, specifically the gold and the purple data. We first did that experiment in one of my Gen Chem summer sessions. So they were very helpful in helping us figure out that this was actually something that was interesting. And then I had some lab students who took it all the way to enzyme assays. Um, for this particular paper, we submitted it in mid-September, and we got acceptance in the journal in November. So we started doing these experiments in July, and then by November it's posted online, which is fairly fast for a publication, but it shows you what you can do. Now, the nice thing about micropublication as well is it's a single figure paper. You don't need to have a complete story. You can have, we've got this cool observation and we did something with it, and here's an enzyme assay or way you could structure it to get these same types of observations. So this is a good journal for those types of small stories that are something that should be gotten out there pretty quickly. It does have an article processing charge of $350, but from what I've heard, they are open to having that waived if you are in a place where that's just not an affordable cost. Um, yeah, so as we're beginning to think about the spring and moving forward, these are the MCC projects, at least according to the website, that are out there. But I want to encourage you also, and I don't think any of the MCC leadership are going to say you have to stick to these 8. I would encourage you to explore and do something that you might be interested in as you're moving forward. And that's there's a lot of excitement, a lot of opportunities. I also want to point out that there are a lot of fundamental areas of MDH biology and chemistry to explore. Certainly, non-cytosolic, non-mitochondrial MDH enzymes, the non-human, non-watermelon MDH enzymes have a lot of gaps in their literature. There are some MDHs that don't use NADH; they prefer NADPH. That's kind of a cool thing. You could look at that specificity. Some MDH enzymes have alternative substrates, alpha-ketoglutarate, lactate, pyruvate, even some others could be substrates of MDH, and that could be explored in more detail. And then, of course, one of the ones that I mentioned earlier, inhibitors of MDH as well. So feel free to explore and think about projects, and then realize that um, there are a lot of opportunities to move forward, and you can make a good contribution to the literature out of your cure and the good things that you all do. So, yeah, that's where I'm going to end this. I'm going to stop sharing, and we can do some questions now. And then again, I'll shut off the recording, and they're going to be off the record questions as well. So if you have a question that you don't mind having be on the record, or we want to bring up a conversation or have a discussion about, we can go ahead and have that. And for those who came in sort of midway through, the idea and the goal behind this session is that we are going to try to assemble working groups that are going to be able to take their spring cures or summer cures and move them towards a publication over the next 18 months or so. And so this is the opening conversation, and we'll have additional meetups hopefully in the spring where we can further these conversations and develop these ideas further. So, yeah, what questions do you want to do you have, or what discussions do you want to have? Well, we've got some time and we can do it on the record.
Chris,
Hi, Chris.
Oh, sorry. Somebody else can go ahead.
Hi. Oh, hi, Pam. This is Sailaja. How are you?
Oh, hi. It's so dark. I can't see you. But it's
I'm
dark
sorry.
here too.
Yeah.
Go ahead. Go
Yeah,
ahead. I can
sorry,
wait.
sorry to interrupt. Yeah, you first. After you.
Oh, you want me to go first? Oh, okay. Sorry, I didn't have my camera on. I'm at home, obviously, trying to do a bunch of stuff. Chris, I really appreciate your presentation, and I guess one particular thing I'd like to comment on or ask more questions about is that critical summer period. So I'm just finishing. We're in finals week here, and I have a big lab report due in my MCC cure. And so I'm reading these lab reports, and they did presentations last week, and I did something a little bit different this semester in that I had two lab sections. We worked with two different phosphomimics, and in each lab section, I had these kind of like super teams where one group was purifying wild type, and then the two other groups in the lab were purifying a mutant, and those same mutants were in the second lab section too. So where all this is going is the data is like all over the place, right? Even though I grew the cells, I made the pellets, they were using exactly the same, you know what I mean? Like, okay, they made up their buffers. There were some things they did that they had to do on their own. But the bottom line is that, I look at this data and there are trends, but oh my gosh, the data is awful. And they realized it when they were presenting, right? And saw other people's. It's not awful. I take that back, but it's certainly not publication quality. And so I guess, I appreciate if you want to talk more about it, that critical summer piece of going back and actually like repeating the experiments is probably super important to feel confident in the data, if that makes sense.
Yeah, and I think you bring up a great point that most of our students have 7 to 14 weeks of experience working with the system, and we can't expect them to put out publication quality data in that time. And I like what you said. You can see trends, and that's how I use a lot of my cures when I'm doing this. Is I look for the bigger trends. Occasionally, I'll get a student who's got golden hands, and I try to recruit them for the summer work if I can. Exactly, but yeah, so I think that having that extra time is useful. And I know that I've been able to go back and pick out some of the key pieces and repeat some of their work in a week or two myself. And so having that extra time and taking that time to do that summer to reproduce everything is key. And I would encourage those of you who can to do that. And if you need extra support during that time, reach out to someone who can help out, because this is also where collaboration can come in to help. Hey, I've got this trend. Did you see that trend? Cool. We should work together on this. Maybe I'll do this mutant, you do that mutant, and try to make it so that you're not spending all summer in the lab, though that sounds like heaven to me. Others may have other things to do during that time. So
No, I really appreciate that. I mean, I'm even discovering in these reports like we took the one that we only did in singlet, not triplicate. And we decided to go with that one, and I'm like, wait, what?
yeah,
I'm like,
exactly.
why? You should have at least talked about that. You discuss. Anyway, sorry, I'm deep in these reports, like discovering new things I didn't realize actually happened during the lab time.
Yeah.
Sorry for that.
No. That you bring up great points, and I really like that you highlighted the challenges that are going to be to take this data forward. But it's an important step. You need
Yeah, I
that time,
appreciate
whether
that.
winter or summer. Yeah.
And Chris, my data is also all over the place. So one semester, I had, I thought what was a mechanism. I mean, I thought I was using a mechanism based inhibitor, and then it turned out that it's actually an activator of the MDH. It's a pretty neat finding. There was a marked enhancement in the Vmax, but I have to do a lot of follow-up experiments. So in my second iteration, I didn't do the same thing, but because I had a little bit more knowledge, I started from scratch from the cells and then expressed the protein. And then same thing that Pam mentioned, making no, I make the cells, I make the pellets, and then I get the protein, and then they purify it and all that. So I didn't, unfortunately, my second iteration, I didn't get to as far as getting some pure enzyme to test it. So again, my data is also all over the place. Um, then I have a grant that I have written that may get funded in the summer. If that happens, then I'll have a chance to test all that. But in the meantime, in the spring, I am teaching a biochemistry lab, and I might have somewhere between 5 and 7 students. So I'm and because of my all other classes, I really don't have the energy to spend an entire semester for a cure. But I want to do a small slice of it, maybe 3 to 4 weeks or something. Plus, I'm on the tenure process, so students don't take, and my department doesn't take kindly if the experiments fail.
Hmm.
Uh, so I'm also running up against that once in a while. So I'm in a dilemma. What's a good way to approach this? I still want to continue my cure work, but not make it my whole semester. Maybe 3 weeks, and I'm thinking of actually doing some modeling studies. um, would you be willing to collaborate with that? You probably have experience in docking experiments and
Yeah, and I certainly have some protocols that I could share with you to help get you there, and
oh,
that of resources that are freely available. They're on the web. You don't need a fancy computer, and you can get answers in an hour or less. And so, yeah, I'm happy to share docking protocols with you.
yes,
Yeah.
yes.
Yeah,
Um,
and happy to consult with you as well as you're trying to design what you're trying to do to help get the students where they are. But I feel your pain, in that it's tough to get these things to work. And it sounds like you're doing the right things. And maybe if you're doing something for 3 or 4 weeks, you might see what has tended to work in the past and really focus on that, even if it's only one inhibitor for the 5 or 7 students in total.
okay, okay. And for the docking studies, I know there's so much literature out there. I mean, even our own website has those resources, but I would like to sit down with someone for an hour and just just to get started with this. Would do you have time for that in the spring?
Yeah, potentially. It depends on what the topic might be. From the technical standpoint, I'm happy to support, and I know a little bit about the MDH molecules, but I also know that there's others who may have some of that knowledge too. So, if you're looking for
Sure.
tech support, I'm there. If you're looking for knowledge support, I'm half there. I can't
Okay.
guarantee that I can fully help you, but I can help you find the right person, hopefully.
But
Okay. Thank you.
I wanted to add to that. So reach out in Slack, and I want to encourage people not just to go to 2 or 3 people all the time. There's a lot of people that even if one person isn't the absolute expert, but together that's how you can do it and spread the wealth, right?
Yeah, and I think one thing I'm going to do in the next couple of weeks, Joe, and maybe you, if you're willing to help me with this, basically with dissemination, is I'll put together some links of things that I do and some videos and make sure that it's all collated in one place that people can find. So if they want to do docking or they want to do modeling work or something like that, that they've got access to some of the stuff that I do. So I know it's a little bit different than others.
Yeah, I think that would be great because some of the stuff we have is a little older that's up there now, and things change so frequently. I mean, almost week to week. Bolts, how many now? So yeah. I had a question for you, Chris. So when I've been talking to a couple of folks about trying to do like one of the micropubs. Now I understand you could stitch a few micropubs together to create like a series of a paper. But what if you had a micropub and then later on you wanted to use some of that data to do a full paper independent that either uses that data or replicates that? Because it's a micropub, is there like a different view of that? What's been your experience, or what have you learned anything like that?
Yeah, so I don't have experience with that situation directly. But you are right. And just to explain how you can stitch the micropubs together, and then I'll get to your answer, Joe, my thoughts on your answer, Joe. Um, Micropublications explicitly states if you want to put together 3 or 4 of these papers into a meta publication, they will let you publish the small pieces and then you can publish another one that is analyzing all of the things together as the complete story. Um, in terms of being able to take the published item and then reuse that data in another published paper, I think you might get pushback from the other journal, the second journal. And that may be where you want to lean on bioRxiv and preprinting your story. You might have to put a little bit more data into the preprint, and it may sit there for a longer time as quote unpublished but preprinted. But then I haven't met, I haven't seen any journal say if it's preprinted, we won't publish it. I think most of the journals have come around to preprints are good because then they direct more eyes to the published paper in the end, and they're different enough usually that things happen. So I would, in your situation, without having the full details, I'd go preprint first rather than micropub. But if you want to string together 4 or 5 micropubs, I think that could work, but you'd have to go back to that same micropublication journal.
So so stringing together and serializing or a meta, the micro, is that within the micro publication? Okay, so then they'll have a bunch of micropubs, and then every now and then they'll have the stitched together meta.
Yeah.
Okay,
It's
well, and
it's
that's perfectly cool.
Yeah, that's less frequent, and I think I would confirm those details with the editor, but that journal's been fairly responsive when you reach out to them. So I'm sure that they'll give you information or point you to the website.
Something else I just wanted to share. You can talk the price down a little bit depending on the journal and how it's going. I had an experience where I was ready to pull my paper because I thought the reviews were bad, and um they started to agree with me, but then I said, well, I'm already looking at another journal which is a lot less expensive, and they dropped their price by half, which was shocking, but I'll take it.
What journal, Joe, are you willing to share?
Um
You don't have to share. I was just
yeah, no,
curious.
I'm happy to share. I'll put it in the Slack later on. I can't remember. It was an NHE paper that I did. And so it wasn't, a JBC or, the only way I'm going to get it in Nature, Cell, or Science is if I buy an ad. Um, but yeah, I'll share it. It was my last NHE paper. I've just got to go find it for you.
We just got a Journal of Chemical Education paper published with open access, and it's just unbelievable what they charge, right? Like
Yeah,
for
well, when
education.
everybody wanted free access, they kind of forgot about the cost of
Yeah.
things.
And to add on to that conversation, I know that the Commonwealth of Virginia library system offers open access publishing funds. And so if I want to publish in a journal, I don't have the money to do it open access, I can apply, and I think I get 2 or 3 shots a year at this, but they'll give me the money and they'll pay up to some thousands of dollars of the open access fee. So you may check out your library, institutional libraries, or your Commonwealth or your state library system, because they frequently have those types of funding mechanisms available. Other questions or discussion topics or related to this?
So, Chris, you said you envisioned that this would be like the starting point for people to be working or collaborating together, or just kind of networking.
I think it can be both.
Yeah.
I see a lot of opportunities. We've got the MCC is fairly mature now in that we've got people who can do the cures and are getting more comfortable with it. There's still rough patches, but I think we're in a position now where the system is working. What can we do now with it? Can we leverage it to start producing research data? And I think that can happen. I have faith in our students. I've seen some really great students do these projects, so I'd like to see if we can offer this opportunity. And this is the infrastructure we're starting right now, is to start assembling these working groups, get these conversations started, and try to make it something that we're doing every year. End of the year, okay, who's going to do what in the spring? And then let's get these conversations going and facilitate the process, and also develop our faculty, get them more experience in that arena of publishing and getting comfortable with it as they see fit.
But to that point, and maybe this is something that should be done in Slack or follow-up chat, it might be good to have like a little, bulletin board or posting of like who's working on what, what's their timeline, and then it might be easier than to know, oh, well, this person's doing that. That'd be cool if we can maybe work together, or maybe their students want to try what we're doing and vice versa. I don't know, so because I think we only have what 5 or 6 people here, and there's a lot of people in MCC. I don't know, like finding the right form to make sure we all know who's working on what. Any thoughts on that?
Yeah, I agree. And that's why we're having this conversation now, is to open that idea and get it in people's minds. And then I anticipate the first week of January, we'll post a Google Sheet or something of, hey, Bernson's doing this, Joe's doing this, Eric's doing this. Is there interest? Do you have ideas? Where do we see these overlaps to start making that Venn diagram of putting people where they can best get what the help they need, whether it's they want to collaborate or whether they just want to follow along with someone who's doing the same types of things and have a buddy, a writing buddy, even if they're doing something totally separate. I know Joe and I have written a couple things, and it's helpful to have each other just to be accountable. Yeah, if I know Joe's going to yell at me, I tend to write faster. So
He does. He does. Awesome. I know I've got a couple of projects after January that I'm going to start putting out there that like maybe the micropubs that a couple of other folks are on. There's going to be room for more. And I think also too, even if you just like you don't have a mutant, but you want to have a mutant that is going to go somewhere, right? In a micropub or a full pub. I think that's where if you see something or if, don't be afraid. If somebody, if Tom puts out, hey, I've got this clone that I'm starting to see some on. Does somebody want to join me? I think everybody's here is friendly enough and we don't have to like be invited. I think we just ask, people can always say no, if that makes sense. If I just blathered because I'm tired too at the end of the day. And certainly don't worry about Joe Ellis or Chris or, Eddie. Just do it. Do it.
Yeah, I mean, I would love to collaborate with someone, and it'll be nice to have who's interested in what. And I think one time, Joe, you had some what's the word? Some topics or subtopics where people indicated I'm
Yep.
interested in this and that. And I expressed at the time, if you remember, I was interested in something, and then you're gonna match me with somebody. I mean, was that I kind of remember up until then, and it might have been Pam. I don't know. I don't remember exactly.
If it was, I didn't know, sorry. Or I forgot.
You don't remember? Well, we'll go back and look, but also don't be shy. Say, hey, I really want to do something, but I'm not sure where. I think there's a lot of space for somebody saying, I got hands, I got interest, but I'm not sure where to go, right? Can I join in? You don't have to say, I have this mutant, I have this data, so then I can join, right? We're all willing to bring folks on. So yeah, like I said, I'm going to get pretty active. I was teaching overload this semester and being chair, so I'm not being overload. I'm not teaching this next semester, and then I go on sabbatical. So unfortunately, you all hear a lot more from me, but it doesn't have to be me.
And also, Chris earlier showed like what you have as an instrumentation at your school and Joe's school. And I'm not sure if we already have this or if it's helpful, but if everyone can indicate what kind of instrument we have, so that if anyone needs anything, maybe ask.
Maybe that might be something where we could ask people to put in, whether it's a Google form of something or even a website, right? Or a link from the website to there, something like that. I think that's a good idea. I know we're running focus groups to capture in ideas for things like this.